This project was realized in collaboration of two universities, Rans University and Moscow State University. The topic of my project, the goal of my project is the engineering of multifunctional pharmacological nanoprobe for optical imaging in cells. During this goal consists of three main tasks. First one is the synthesis of quantum dots with advanced photoluminescence properties for biomedical application. Second is the synthesis of new DNA ligand capable for conjugation with the quantum dots. And the last one is assembling of multifunctional nanoprobe. In the slide you can see principal structure of our nanoprobe, which consists of pharmacological agent capable to interact with single-strand or double-strand DNA, vectorization group for delivery of this system into the cells, and also imaging component, quantum dots, which is responsible for visualization of our system. My project consists of three main parts. The first is the synthesis of quantum dots. Second is the synthesis of DNA ligand, and the last one is the assembling of nanoprobe. Quantum dots is the main topic, so I'll start from it. What is quantum dots? Quantum dots is the semiconductor nanocrystals with photoluminescent properties, which can be easily tuned by changing of size, composition of the nanocrystal.
Вы не можете микрофон свой выключить, потому что те, кто у вас говорит в комнате, все здесь слышно очень громко. Вы слышите меня, да?
Да.
Ага. Попробуйте перекрыть микрофон, а то все люди, которые там в комнате, мы их слышим, которые говорят за спиной у вас. Окей.
Okay. It means that by changing only one parameter, size of quantum dots, we can tune photoluminescence of our nanocrystals in a wide range of the spectrum, starting from ultraviolet to infrared. So during this project I used only cadmium selenide quantum dots, because this material is very useful because of big ability of precursors, which can be used for the synthesis, and the useful region of the visible part of the spectrum. Another main properties of quantum dots that independent of the size of quantum dots, the surface properties of quantum dots doesn't change compared to the organic dyes, which is alternative material for the imaging. Each type of dye has absolutely different chemistry. So in this case, quantum dots is more useful because of the same properties of the surface. Also we can see in this part of slide that photoluminescence band is very narrow. It means it depends on this property depends on homogeneity. So quantum dots can be synthesized with very narrow size distribution in the ensemble, less than 15%. Also, quantum dots absorption spectra of quantum dots is continuous, so it gives flexibility to excitate different size quantum dots. Continuous and very high, as you can hear. So it gives possibility to excitate different size quantum dots by using only one source of light, for example laser. this property with wide photoluminescent band give us opportunity to use this system for multiplexing when we modify surface of our quantum dots with different biomolecules, and after the system can be targeted in the cells, for example, and under the excitation we can see emission light of different quantum dots. One of the last properties which I am talking about, this is this high molar extinction coefficient that gives our high brightness of our quantum dots. Compare high brightness. So, and the last advantage of our QD is possibility to emit into photon mode. So these properties give us opportunity to visualize our nanocrystals from the deep depth and with high resolution. That's very useful for imaging, for example, in vivo. Now I'll talk about physicochemical aspects of quantum dots. So, after our quantum dots can absorb any light. Under this process, there is the excitation of electron and formation of hole on the space of electron. This pair, there is a charge carrier, electron-hole pair. And during the relaxation of this system, there is two possibilities: radiative process and non-radiative process. Of course, if we need to obtain bright quantum dots, we need to increase possibility of the first process with emission of light. So for this goal, typical cores covered by protection shell, which increase localization of our charge carrier in the core. And this shell gives us increasing of photoluminescent efficiency, or also called photoluminescent quantum yield. The best quantum dots typical sized using colloidal approach, using organometallic precursors. This type of synthesis gives us opportunity of wide range of precursors, which is very useful, is very important to modification of reaction conditions. Also, it gives us a possibility to tune properties of quantum dots. During the realization of my PhD project, we choose 6 main parameters which can influence, which has a major influence on our properties, or final properties of QD. So after analysis of this parameter, we choose optimal condition, which are present in this column, which allowed us to obtain universal method for the synthesis of quantum dots, which can be used for, which allowed us to obtain quantum dots with narrow distribution, as well as allowed us to tune, without changing of this procedure, tune size of quantum dots in all region of the size. So this basic condition allowed us to synthesize quantum dots with diameter starting from 2.5 nanometers, with quantum yield close to 35%. But for the biomedical, So during the optimization of reaction condition, so in previous slide I don't speak a lot about this manipulation, because there are a lot of experiments, and if you want I can briefly talk about it in the presentation. So during our optimization of our synthesis condition, we found that composition of reaction mixture has major influence on size distribution on QD. For example, here in slide you can see that in photoluminescent spectra there is two minor bands which correspond to the alternative ensemble of quantum dots. This is the result of unreacted precursor and side reaction with side process. So, in literature, the most popular method for purification of core solution is the precipitation and the redissolving of our quantum dots. But in some cases, this method doesn't allow to make high purification of our system. So we propose alternative method which is not very useful by our colleagues. This is size exclusion chromatography using polystyrene cross-linker beads, which are filled in our column. Finally, after purification, we obtain high quantum core of quantum dots with high purity. This method is size exclusion chromatography was used during all stages of purification, and it gives perfect result. So, for biomedical application, we need to size of quantum dots is a principal parameter for biomedical application. Typical synthesis approach allowed to synthesize quantum dots starting from 2.5 nanometer in diameter. But in case if we need to move from in this part of kinetic calibrating curve, the speed of reaction is very slow, and there is a small quantity of precursors. So if we need to obtain minimal size quantum dots, we will need to move to this red region where rate of reaction is very fast, and there are a lot of quantity of unreacted precursors and ligands. So we propose for the synthesis ultra small quantum dots, we propose method which is based on combination of fast termination of reaction using liquid nitrogen and size exclusion chromatography. Finally, we could obtain quantum dots with diameter close to 1.5 nanometer. This is extremely small quantum dots which consist approximately near 60 atoms of cadmium and selenide. For purification, we use the size exclusion chromatography, which gives us well results. Here in photoluminescent spectra, we can see that that photoluminescence of purified samples is more narrow compared to unpurified samples. Which, as I said before, this parameter influence on size distribution of quantum dots. So this result is one of the main during my PhD thesis, and also based on these ultra small quantum dots, we engineered our multifunctional nanoprobe. So next slide. After we would like to check for the shell growing. We need to know, for the precise shell growing, we need to know exact precursor quantity we need to add to our reaction mixture. So in literature, all models based on a lot of models based on spherical model of quantum dots, but sometimes it doesn't correspond to real result of real imaging, imaging. So in this system, when our model is spherical, it's difficult to understand what's the real structure of our shell. So we proposed alternative model of nanocrystal growth which based on real crystallographic, real faceting of our nanocrystals, as well as crystallographic data which correspond to material of our nanocrystals. In this case, it's cadmium selenide. So we can see that typical shape of our nanocrystal is hexagonal B pyramid, and based on it we built our atomistic model. This model based on several steps or several principles. The growth of nanocrystal, we start from the single monomer, and then we fill our coordination sphere of our monomer by another molecule. For example, for cadmium selenide, the crystallographic number is 3, which means that to each atom of cadmium or selenide, next step we add 3 atoms of material. So, and step by step we continue growth and collect structural parameters of our nanocrystals. So we obtain data, we obtain results, we obtained composition of our nanocrystals, how many atoms of each components, and data which and diameter of nanocrystal. So finally we decided to compare our results to literature data, and here in the figure we can see that our experimental point correspond to literature data and experimental results of calculation. I think it's well result which proof of concept of our model nanocrystal growth based on real crystal which based on real faceting of nanocrystal. Next step I'll talk about we try to use our model to synthesis of core shell quantum dots. So there are two main approaches to increase the photoluminescence quantum yield using covering shell. The first one is maximal increasing of quantum confinement of the shell, and the second is increase of shell thickness. For biomedical application, it's not useful to increase the shell thickness because we need to obtain minimal small quantum dots. So this methods realized in several typical structure of core shell quantum dots, which based on covering of core with zinc sulfide or cadmium sulfide shells, or maybe in combination of these materials. Also, in this structure there is one important limitation, that during shell growing we see that our photoluminescent quantum yield decrease after one of the point, because of there are several reasons of this decreasing. Most of them depends on the structural difference in crystallographic parameters of core and shell. So we proposed a new model of core shell quantum core shell structure, which consists of which consists of 3 monolayers of zinc sulfide, 2 monolayers of zinc sulfide, first and the second, and intermediate layer of cadmium sulfide. This combination of shells give us growth of increased charge card localization because of the maximum quantum confinement potential. And what's the important properties of this shell for the maximizing of photoluminescent quantum field? We need to have that each shell will have minimal thickness, only one monolayer of material. So for the synthesis of this structure, we use a combination of two approaches. The first one is we use data of our models, which allowed us to calculate precise precursor quantity, which we need to synthesis of each shell of each layer of shell. And the second is a cellular approach, which allowed us named successive ion layer absorption and reaction. And this procedure allowed us to precise control of materials, precise control of shell growth. In this method, small quantities of each precursors adding into our reaction mixture and in the thermal cycling mode in the low temperature. After it, we heat our reaction mixture and our precursors start to absorb and react with the surface of previous core. After we repeat our injection at the cold temperature. So this approach allowed us to synthesize different batches of quantum dots with different color photoluminescence. So here we can see that narrow band of photoluminescence and high uniformity of nanocrystals. If you can see that it's less than compare previous core which we used for the synthesis. Also we analyzed photoluminescent efficiency of our structure. So in the figure we can see that during the shell growth, photoluminescent quantum yields doesn't decrease, and it's close to maximum value 100%. It means that our conception is right, and our energy structure of shell materials allowed us to obtain maximal localization of charge carrier in our. Also, we analyzed two photon properties of our nanocrystals, and we found that the main parameter of efficiency of this two photon emission is Geppert-Meyer. The value of this parameter is more high compared to organic dose, and comparable to typical structure quantum dots. So we use this multi-shell structure to obtain the quantum dots based on ultra small cores. So we cover our minimal size core with the same composition of shell. In the result, we obtain nanocrystals with diameter close to 3.5 nanometer and photoluminescence efficiency near 70%. This batch of quantum dots was used further for the engineering of our nanoprobe. So, and in the result of the first part, using combination of optimizing of synthesis properties and modeling of quantum growth and modeling of quantum dots growth, we obtained nanocrystals which has high uniformity and quantum yield close to 100%. So also we obtained with small quantum dots, which we used for the synthesis, for the engineering of our nanoprobe. Next part devoted to the synthesis DNA ligand for interaction with DNA sequences. Telomeres is the region of single-strand or single-strand DNA sequences in each end of chromosome, which protect our structure for deterioration, DNA sequence for deterioration. Guanine-rich end of guanine-rich sequences can form G quartet, which also form planar structure, which are very stable and play important role in stability of our DNA. This terminal structure of DNA sequence attractive at our attention because it plays important role in cancer. So in most of cancer we can see high activity of enzyme telomerase, which plays important role in replication of our sequences. Also, for the stabilization of this part, for the stabilization of G quadruplex, small molecules based on polyaromatic heterocycles can be used, which have high affinity to stabilize, to interact, to fold this planar G quadruplex and to stabilize them. So, there are a lot of potential ligands based on polyaromatic cycles. The most interesting, for our opinion, is acridine-based derivatives. One of them is 3-substituted derivatives of acridine, and another is 2-substituted. Why we use this structure? Because BRCA19 is one of the potent molecules with micromolar inhibition activity of telomerase, and also our colleagues from the University of France, Champagne-Ardenne, have a big experience in the synthesis of these molecules. For example, they synthesized another 2-substituted derivatives of acridine, which also has the same efficiency. So we combined to this structure these two molecules, and here we can see our goal molecule, which consists of acridine core, which is modified with aniline, 3 different aniline derivatives, which allowed us to increase interaction with DNA sequences, phosphate group of DNA sequences, as well as there are 2 main symmetrical arms of polyamine, which allowed us to which play a role of vectorization group in this system, as well as allowed us to make conjugation with quantum dots. So these molecules was synthesized in 7 steps. 3 steps is well described in literature. Another 4 were synthesized at first time. Starting point is acridine. After realization of well-known stage, we obtain 3 substituted acridine derivatives, acridone with oxidized in position 9. After we make introduction of chlorine substituted in this position, As well as we introduced two protected one side by Bohr group, putrescine sidechain. So finally, we introduct different aniline derivatives in position 9, and the last step we made the cleavage of the Bohr protection group. In result, we obtained 3 molecules, which overall yield close to 10%. And next step, for one of the main problems during the synthesis of these molecules, it was purification, because these molecules has not efficient stability. To increase this parameter, we transform this molecule to the salt form of hydrofluoric acid. It allowed us to isolate enough stable molecules. After it, we also will perform purification with HPLC, and after make characterization of basic optical and other physicochemical properties. Here you can see one of this molecule, properties of this one of the molecule. You can see enough high purity of this molecule in the sample, analyzed sample. And also this molecule further was used for engineering of nanoprobe. So